We start the final session of the day, which is databases and biobanks, which will be chaired by Katja Kobov and Sanjay Sisodia.
Yes, welcome back to the last session. So I will give you a short overview on the bottlenecks and problems or issues that we have to consider when we talk about biobanks and databases. And first, let's say something about biobanks, what we need to consider. So the problem with most biobanks that we have is that they have grown from a very small size and now are increasing, and like anybody also in industry who has ever known a company developing to a certain size, they are facing several problems. So here we have issues of space, costs, and now newly with the GDPR regulations that's now in place, we also always have an issue with the data protection that we have to control. Then one thing is always the quality of tissue that we use for our research. So we want to make sure that it's well characterized and is of good quality, because rubbish in means rubbish out. So this is something where we have to maintain high standards and where we have to agree on standards that we apply to the tissue that we use. Also in terms of classification and everything, it's not possible. I think it's a critical issue that taxonomy is something I will mention also in the context of databases, that we speak the same language and also adhere to classification schemes introduced by the International League Against Epilepsy. A big question is what type of tissue should be actually collected. So we have been collecting blood, which we use in genetic studies, but we also have access, and that's very unique in epilepsy research, we have access to brain tissue from focal epilepsy patients which undergo surgery for treatment. But then the problem is we're now moving into more advanced analysis where we look at single cell populations. So the question is, should we actually start to collect prospectively individual cell populations? This adheres to brain tissue and also to blood. Then should we start to collect fibroblasts? Should we start to collect saliva from every patient? Is that useful or is that not useful? And how can we justify that with the ethics? And how can we then again talk about space and costs and everything? That is a big issue. Then, of course, the limitations in access are a big issue. So here also we have realized during the last 5 years that ethics is a big problem, harmonization of ethics between European countries. So something that works in one country doesn't necessarily work in the other country, and that actually limits our perspectives for working together and doing research together. Same is true for material transfer agreements, and this problem is not only within Europe, but gets even more complicated when we try to work together with other groups from the States, where again, completely different regulations adhere, and that's a problem. And then joining biobanks and databases, that is also something we need to consider. Then talking about big data and moving to databases. Big data is an issue, of course. I mean, epilepsy is a complex disease. The brain is a complex organ, and the type of data we generate is really large. Whether it's EEG data, imaging data, or omics data, or whatever else you can imagine, storing all of this is a challenge. Gaps to be filled in the context of databases is first of all, again, the taxonomy and ontology that we use, so that we use the same words to make actually bioinformatic analysis of our data possible. because when we do use different words for the same thing, the computer will be confused, let's say. Then a big issue that has been discussed in the last years is common data elements. So what kind of data are we actually collecting in our databases? What is a minimal data set that we should collect from every patient? How do we standardize that to make the studies that we envision actually possible? Then there's a big challenge in data transfer for storage and transfer to researchers. Which starts with the technical infrastructure at each site. So do we have the computer systems that we need to actually transfer these big data volumes? Then there's the safety issue when we talk about where do we store the data, on local servers or commercial clouds, and who gets access. And yeah, again, adhering to GDPR is very important. And then one issue that I want to mention, as we heard it throughout the day, it is a little bit like the current state is that with every new project that is launched or every new consortium that is launched, a new database is created. So this is like the first thing every consortium does. Then after 5 years, the project is over, and then who's going to maintain that database? Who will actually pay for that? And then, you know, most of the time things get lost because nobody takes care of it, and this is really a pity because we put a lot of effort and time into this. And how do we actually can come over this specific problem? I think this is something we should think about. And then, of course, we have different databases for different data, but in the end, we would be interested to join all these things together. And how do we do this if everyone is using a different platform? Yeah, I mean, there's a lot of issues that needs to be considered. And with this, I come to an end and hand over to Ingmar Blimke for the first talk. Thank you.
Thank you. Thank you, Katja. Thank you, Katja. Welcome, everybody, to this last or late evening, late afternoon session. It's my pleasure and honor to talk about the success of the European Epilepsy Brain Bank despite all these challenges and bottlenecks that Katja just characterized. I will give you a very short introduction into the history of it. It's almost 12 years now when we started at the FP6 consortium called EpiCure, at that time launched by Giuliano Avanzini. We had a work package there, and we thought, no, let's start this European Epilepsy Brain Bank project. And it was first started and considered to be a virtual database. So what we just did, and Katja just mentioned that because we were at the front of that development at that time, we started this virtual database by defining a minimal data set that were just 8 items that we thought characterized the patient well, and it is histopathology based. So we collected the pathology diagnosis. In other words, this is only patient that went through epilepsy surgery with the intention to cure epilepsy. So epilepsy resection to cure epilepsy. So no like callosotomies or other palliative approaches. So histopathology based, then we just said the patient should be characterized by its age at onset, the duration and age at surgery, then the location, meaning the anatomical location of the resection, the site, left, right, the sex, and that's it. That's a very small data set. And over the years, we continuously we grow continuously, and then with the FP7 consortium, the Desire Consortium, we became also a biorepository. So we were allowed to store tissue, and that was because we had that this clinical trial of Helen Crosses where we thought we should be flanking and supporting that clinical trial by collecting the brain tissue and going for some molecular studies underpinning and underlining the hypothesis. However, that was 2013. Now, today, we have more than 10,000 patients collected. The minimal data sets of them were published last year in the New England Journal of Medicine of 9,523. So these were those, they're almost completely, the data set was almost completed. And with the let me see if there is some, yeah, you see this was EpiCure, you're missing the logos. And now with the EpiCare, we further develop into an reference platform for digital pathology across European countries. And I hope I will have some time at the end to show you our approach. So these are my results. The yellow countries spot the countries, 12 countries that contributed to this approach. There are 36 red pins. These are representing the centers contributing. And as I just said, we had now more than 10,000 of these patients collected, and 9,523 of them were published last year. And that is one of the graphs, and I really cannot go through all this data. It's obviously a very large data set and gives us a nice overview, and what I would say here as a summary, a catalog of those brain lesions that are amenable to epilepsy surgery. And it was a total of 36 histopathology lesions, and interestingly, the top 10 of them almost covered 80% of the entire cohort. That's very robust, very solid data. And we classified them into 7 major disease condition or categories, and you see that they are very, very robust numbers in here. However, I would like to draw your attention to the onset and duration, saying that more than 3/4 of all patients had their epilepsy onset in childhood. Now, onset is mean here is 12 years through all the categories, with the exception of the cavernomas and so forth of the vascular thing. However, the duration of the disease before surgery was indicated or was performed was another 16 years, 1/6, so almost 2 decades. I think this is a big number, and this is where we have to work on. So 16 years of frustrating anti-epileptic drug treatment with all that in a child. You see, and then we had also collected secondarily the outcome. This is a 1-year outcome, complete seizure-free, complete, no auras, no seizures at all, ILE class 1 or Engel class 1A. And however, the New England Journal did not allowed us to publish that data looking for the predictors, because that was not our, was not pre-specified when we start up the project. So now we call it the Epilepsy Brain Bank 2.0 project, where we started to recollect all the data again as pre-specifying, and we are pretty sure that we can identify also some interesting predictors, but that is for our next year. So the bottlenecks that just was that the bell already? No, this was Helen. Okay, sorry, Helen, sorry, go ahead. So what I just said, the definition of the data set is critical. It has to be predefined, data safety, encryption, storage. This is all what just Katja elaborated on, ontology and language issues across European language. You must believe me, I have probably 12 different words now for hippocampus grossus in my mind. That's something we have to come along. And the classical Klaus-based solution of centralized databases, I think this is not 21st century style. So we have to rethink how to do that better in the future. So kind of this MyCloud solution, where the patient himself carry all the data and has an app and can, respond to any request from the consortia. So the future is, and make a very strong statement here, what we need really is big funding. So I make a prediction: if you give me tomorrow, or the European Union would give me 10 million euros tomorrow, we will have a new disease classification in 5 years from now. And I can prove that by the a huge success in the field of brain tumors over the last years. This was all tissue-based in the brain tumor field. They come up with really exciting molecular genetic, pathological genetic data, and even the second March issue in Nature, we published together with these huge consortium of the German Cancer Center in Heidelberg, an FFPE based, so formalin fixed paraffin embedded tissue, DNA methylation approach, and that revolutionized the whole WHO classification of brain tumors. And we can do the same. We have these 10,000 specimens. They're all paraffin fixed. There's no business. It's just missing the money and manpower. And then we could do the pathology genetic disease classification. Now we introduced digital microscopy solutions into that and become a reference platform, pathology platform for the EpiCare Center. And of course, the MyCloud patient-centered solution I just mentioned. So this is my last slide here. This is the workflow that we think, where we have a digital pathology available, and that should be also introduced into the databases of supported or supplied by the European Union, because this histopathology fingerprint is like a genetic fingerprint. It's very specific, and we touch only the surface so far with our naked eye on the microscope. When we do that on a large, deep learning algorithm way, I think we can receive much more information about that. So that's the end of my talk. I think I stayed almost in time, and thank you so much for your attention.
Thank you very much, Ingmar, for showing us what biobanking success can look like. So our next speaker is Dr. Anand Bishop, representing the Epimyrna Consortium. Thank you.
Okay, so first, what does what? So this is the thing that moves, and this is the pointer. Excellent. Okay, can you hear me? So yes, first of all, thank you for inviting me here to give this talk. I would first like to emphasize these couple of issues that were brought up in the introductory talk about how people just don't connect with their needs and with what they provide. And today I have had a couple of talks with total strangers to me. And there are clearly groups here. Every single one in this room is probably either looking for patients with certain kind of data or has those patients with certain kind of data. If I ask you to raise hands in either group, I would say it's about half and half. So guys, talk. You are here for that. So I, as a complete stranger and outsider, can immediately locate people who could just pair up and have a little chat. So you can do it as well. This is also for the coordinators, by the way. You have already deep knowledge about this, I'm sure, but make them talk, right? Okay, I'm not consuming more of my precious 8 minutes here. How does this work? Oh, it's an animation. All right, sorry. I'm from BC Platforms, a company that builds clinical genetics databases, and we have recently started also working in the fields of omics, and that is the reason why we are part of Epimyrna project. And the mandate for this project was to build specific resources for Epimyrna consortium. Oh yeah, the text is there, I can see it from here. And some community dedicated resources. Part of this work that I'm going to show here is actually totally done by others, and some part is done by our company and also the people working on the platform that we built for them. And so I'm not claiming to be expert of all of this information here. In fact, if you have questions about the scientific part of this, I will just simply point fingers at the audience and pick the person who knows about it. So it is all about work package 6, where we were supposed to build a data storage sharing platform for bioinformatics and multi-omic dataset integration purposes. And also in the same project, there's been quite a big development in getting Epimer base running, a website there. Please go and visit it. I just got information from David that we've already actually, or this page has now reached the magical number of 5,000 individual visitors. So it's going strong, growing. It has about 2,000 plus entries of different information points about microRNAs and nice links to outside evidence as well. Studies published during this project. And the omics platform is a mixture of patient data and rat and mouse models. Most of it is revolving around RNA expression, and then of course microRNA identification, enrichment, expression, and so on. And I'm also mentioning here the iClip methodology, of which there was a nice poster today. Morton knows more about that there. And it is quite a nifty technology for collecting microRNA targets. I'll mention that probably also a bit later in this. So here come the slides that I don't have such a deep knowledge about. These are examples of how the data, omics data, has been generated in the platform, so what kind of experiments are included in these datasets. So here we have the microRNA profiling work, which was also described. Parts of these were actually described in David's talk earlier, and parts of these are in 4 or so posters in the poster session room. I hope you have seen this as well. So basically it is a hippocampal compartments expressing different kinds of microRNAs. And in these models, they pull together microRNAs that are common to all of these models, and continued screening from there. And this just illustrates different time points observations for each of these microRNA types. So basically you can see here what kind of information can be actually generated and stored from these kinds of experiments in the database. Equally here there are some more examples about different points and quality and metrics, statistics collected. And specifically this, I think, was mentioned by David, how the different microRNAs are enriched in different parts of the hippocampus when the hippocampus is actually dissected and analyzed in compartmentalized manner. Okay, so basically everything revolves about the interaction between messenger RNA and microRNA, where this iClip technology has definitely proved to be quite useful. So it's basically a manner of pulling down enriching microRNA, which at the same time pulls down its targets in the affected tissue. And this allows you to analyze the messengers and the microRNAs at the same time, pinpointing certain seed nucleotide sequences in the messengers themselves, and you get a pretty good chunk of data for analyzing these two together from specific affected tissue sample. Okay, the database architecture a little bit here. So everything revolves around the data warehouse, which allows you to analyze these different omics datasets, store them, share them in different kinds of fashion. They can be public for all the users in there. They can be protected so only certain people see them. They can be totally private. It's possible to put applications on top of this for bioinformatics analysis. It is possible to share also the data outside the system, if so is wished, and it is possible to build data input pipes that feed the system with more data if such pipe exists. And that's just a couple of screenshots of how it looks, so what you can do in there. So I got the summary of Epimerna's achievements. So the Epimer base is available. Please go and click around. We have the microRNA database there and the multi-omics atlas for the consortium internal use at the moment. So this is again something to discuss in the future, how we make these available for everyone. Hopefully at least in this collaboration event. It would be nice to have some suggestions. And the challenges are pretty much the same as with any database development project. So how to curate and annotate the data so that it is harmonized in the same way as many other datasets are, follow other standards to follow. Where can we find more information about the actual datasets? What were the experimental conditions? How are we supposed to find more information about them? How are we supposed to filter data belonging to certain kinds of experiments and so on? So these are all about the database design, but also about the diligence of the researchers to provide this information so that it can be used in a useful manner. Funding, collaboration, and the long-term supports are always, as was mentioned before, issues in these kinds of projects. So how to provide for the future, really. And the next steps in this specific project are, again, about data curation and annotation, so that can always work better. There are always improvements to be made. And access solutions, community tools, that's definitely something that we need to look at, so how to share it in practice. Thank you.
Thank you very much. I think you make an important point about consortia working together. We should look to see how we can do that more. So our next speaker is from epiPGX, Rollin Krauser. Thank you, Rollin.
Okay, so thank you for the invitation of presenting the data management part of epiPGX here. So, okay, epiPGX has been presented before, but I'd like to stress the fact that the consortium is pretty interesting in its composition, in that many of the groups contributed samples as well as did analyses. So we had to build an analysis platform fairly early on. So I'd also like to stress the importance of the British groups, like four groups contributing, and we hope because we are still collaborating and hope to do so for a longer time, that the Brexit won't throw us any sticks between our legs, and that the jingoism on either the UK or in Europe is not going to play havoc with this collaboration. So the data set that has been compiled is pretty impressive, I find. So we have about 12,000 cases collected in the phenotype database, with 40,000 AD trials recorded. So some of this data was prepared before, was converted from existing databases and then augmented. You can see in this graph up here that some of those jumps are these points where we introduced patients, and most of the cases were genotyped. Genotypes were also contributed by projects from partners that were collected before epiPGX started. There's a standard set of controls. You can, of course, get more controls from dbGaP. Kind of serendipitous in the project was that we had the capacity to sequence about 3,000 exomes on top of this. These are all cases, and both resources contribute quite a lot to international projects, such as the ILAE Consortium on Complex Genetics, as well as the EPI25 Consortium, and certainly the experience that I got from working in this group also helped these international consortia and to build CRFs and the like. Okay, so let's talk about research infrastructure. This was mentioned even this morning, and of course we have a high-performance computing infrastructure, and I could spend the rest of the talk discussing the numbers of CPUs and how they're made, but that of course is not very interesting to this. What's actually a lot more interesting is that you have a high-performance computing system that you can actually use, and I'm quite happy to hear from our partners that we have a usable system, because I guess every university that is doing research has some kind of infrastructure in place. And I believe that you also need somebody who's doing what is now called data steward services for a consortium. That's a term that's fairly recently introduced. I've been jokingly referring to myself as a data janitor for some time. I guess that's a step up. Of course, we have separate. I did not include any block diagrams on how the database is composed and the like. But let's say we separate data by types, so the phenotyping data and the genotyping data, and are managing access to that consortium. So you need somebody that you can trust. And I think that's actually a lot more important, that there's trust between the people who are working than having the technical solutions in place. So the technical solutions that are out there work better or not. Most of them are growing a lot over the last couple of years, and it's a very active field. I'm also involved in one of the bigger bioinformatics infrastructure projects, Elixir, where we see what's going on in domains outside of epilepsy research and outside of any kind of clinical research, and you realize that there is a lot of development still that needs to be done. I think it's important for a good data steward service to be integrated with the platform and to work with the analysts. I see now that people start hiring data stewards to take care of their data. I think that's very difficult if you don't know how things are going, and I can talk from my own experience, because when I started I was a yeast biologist or bioinformatician and had very little, but that's some time ago, I think. And I'm very happy to have this multidisciplinary team there that helped us to get off the ground. So it's so here's the screenshot of a CRF and or HPC structure, which I've actually never seen myself because the room is not available to visitors anymore. That's there. So one of the things I would like to take to take home from this is that there is now a style guide, if you want, for data management called FAIR. That stands for findable, accessible, interoperable, and reusable data. And it's kind of a suggestion, a broad suggestion how to do data management and data stewardship. And if you haven't heard about this and your ears aren't bleeding yet, then do take a look at it and maybe read the book, in which you will find that all of these terms are not really hard defined. There's no hard definition behind those, right? So findable does not necessarily mean that the data is sitting openly on the web, as some people have feared. Of course, we're dealing with sensitive data, and it's more that the data needs to be standardized and organized, right? Also, much of the FAIR data management is talking about post-publication, but we really need to take care that data is managed before in the project and not just after like the 5 years that you might actually work on one of those projects. Nothing's free. This slide you certainly know. It's the cost of the genomes falling, and up there is Moore's law, the law that says that computers basically double their speed every 18 months. The problem is that, for instance, storage is also there, and that actually does not grow as fast, so we're really running into capacity problems more than we're actually running into CPU problems. But something that was mentioned briefly by Holger Lerche this morning, the phenotyping actually now gets really costly, particularly if you're working in a field like epilepsy, where you want to have a trained neurologist who's taking care of data entry, at least reviewing things. So, and in epiPGX, we have like something like 150 variables, key variables in the CRF that does not, and if you're doing this retrospectively, as we've done, that's a real cost. And as you know, some people start to throw away data rather than, and resequencing them rather than storing the data. You can see that data entry is going to be a real challenge. So maybe the last minute on 4 points that what we can do. Ingmar Blümken said the centralized solution cannot stay. This is a concept that will not stand, and I absolutely agree. I I'm not 100% sure that we're going to manage to have patient-centered data very soon. I think what we're going to have, at least for some time, is something that's called a federated research infrastructure. That's a term that people throw around, where data is centralized in some sites that are kind of as a steward to this data set, that data organization works across those sites. There's a lot of interesting research being done on encrypted data sharing, so rather than leaving this data, you stay at the hospital or the research infrastructure locally. Nothing of that things is workable yet. Nobody will currently be able to introduce this and run it, but it's something. There's a really interesting paper by Bonnie Berger just out in Nature Biotech, I believe, that actually promises to actually do GWAS in an encrypted fashion. If that really comes to play, then a lot of things will change. Data management plans are a really good tool. You have to build it. Thank you. You have to build it anyway if you do an H2020. If I have more to say, I'll go over it. We need to improve phenotyping and data exchange, and one thing that at least I can do is run a course on it. So that's the last thing I would like you to take care of this. So if you are, so this is HPO based with Peter Robinson, the head of HPO is actually going to be there. We talk about EHR and how to extract the most of it and how to actually do your databasing right. Course is open for registration now. Thank you for your attention. Oops, sorry.
Thank you very much, Roland. So next is Eleonora Aronica from the EpiStop project.
Don't touch, but I touch. Thank you. We are almost at the end of this long but interesting day, so I promise I will be brief, but I would like to share a few thoughts and to address few issues about brain biobanking. So we go back to the brain, to the tissue, and as a basis for actually what we wrote here is the translational research. So biobanking is, first of all, I would like to introduce biobanking, keeping in mind that biobanking is not only include surgical tissue, but involve also the collection of a post-mortem brain tissue. And then I will use tuberosity complex as actually model disease, just to show briefly some results which have been obtained using human tissue within EpiStop, but which represent also the basis for interaction, and I will show there is a real interaction between consorts, and then some challenge. So biobanking, we all are aware that the public awareness towards the research biobanking is really increasing because we are in the era of personalized medicine. Actually, the patients are becoming more and more aware of the importance and the benefits of tissue biobanking, which is part of the really routine infrastructure for personalized medicine. I think we have to consider this. Why biobanking or brain tissue biobanking for epilepsy? Because we have a lot of models, we just hear today, and genetic model, acquired model, but they are still models. So we need human tissue actually to validate this model, to prove the translational value of this model. So we can use human tissue to validate the data of animal model, but also to generate a new idea, new hypothesis. So and then I would like to really point out the value of the human tissue and collection of post-mortem human tissue within the spectrum of epilepsy-associated disease, not only for tuberosity complex, but also for other types of epilepsy which have been studied, for example, in other consortium as EpiTarget. And clearly, if we want to understand the pathology of the brain of patients with epilepsy, we have to have some control tissue, good quality. So I would like another time to point out the value of a healthy brain biobanking. And study developmental disorder, as we do within EpiStop, we need to get inside in the normal brain development, maybe going back to the fetal brain that we use also within EpiStop. So another time, I would like to point out the value of, in this case, brain biobanking. And then surgical post-mortem tissue can be used also to obtain and to study selected type of cells. For example, we'll use the human astrocytes in our study, but also human neurostem cells. So tuberous sclerosis complex has model disease. We just heard a lot about this genetic disease due to a mutation of these genes, which actually what the cause is a deregulation of a pathway which is important during development, mTOR. So it's not a surprise that the characteristic of patients with TSC, they are the brain lesions, because there is an alteration of corticogenesis. The tuber, which are epileptogenic, which explain epilepsy, but there is more than epilepsy. We just hear about comorbidity, about autism spectrum disorder in these patients. And the availability of post-mortem tissue offer the possibility, as it did, to look outside the single lesions, which can be resected at surgery. We know that in TSC brain, all brain is affected. They are not only the tubers, the macrotuber, but small lesions. So there is a micropathology which is affected the gray, the white matter, and which is also dynamic. And the availability of fetal brain offer the possibility to prove that these lesions are already present in fetal brain. Already at gestational week 20, you see a tuber, you see that there is activation of mTOR, so we can already conclude, and this is using actually the post-mortem tissue, that there is a pathological network with cellular and abnormality, molecular abnormality, which explain why seizures in this patient develop during the first really months of life. So I think tuberous sclerosis complex, I believe, is a unique disease to study epileptogenesis in a relatively short period of life, a well-characterized monogenic disease. It clearly is an epileptogenesis related to a specific pathway, the mTOR. And then you can say, okay, mTOR is all involved in TSC, it's really a small population of patients. But actually, mTOR, which I can show here in its complexity, is a pathway which is dysregulated also in acquired type of epilepsy. And we know, and we just mentioned, that it's also involved in a subset of malformational cortical development which share common features with TSC. So actually, these represent the basis of the collaboration which has been established between EpiStop, EpiTarget, and the site. So a few examples, we use human tissue to prove indeed that there are similar histological patterns between, for example, tuber, focal cortical dysplasia type 2, and there are similarities also in terms of alteration of the maturation during corticogenesis. We also use human tissue to study the pathology of the white matter, which is important to explain the link between dysregulation of mTOR and comorbidity, showing, for example, that here we are dealing with this maturation of oligodendroglial cells, actually with insufficient turnover, which is still linked to the mTOR, but also to other factors as inflammation. And then we also use human tissue to study the pharmacology, the function of the native receptor using Xenopus ovocytes as expression system, showing, for example, that GABA or AMPA receptor, they have immature properties, and this is also common in a spectrum of malformational cortical development. And then we use also human tissues to get inside in the global change in gene expression, looking at the coding and non-coding RNA, for example, highlighting between the coding RNA the upregulated genes which are related to inflammation. So another commonality with other type of epilepsy, alteration of innate immune response. And also you can use human tissue, as Kasia already showed, to study epigenetic modulation. In our case, we looked at the interleukin-1 beta. And then we looked also at the non-coding RNA, highlighting a family of microRNA that is the 34, which for us was of interest because it targets genes related to neurogenesis, and it has been associated with autism spectrum disorder. But this represented also the opportunity to establish a collaboration with colleagues of EpiMIR and then to study more the function of these specific microRNA. And then we also study inflammation, and we use actually human astrocytes to identify 2 microRNA which are induced by interleukin-1 beta, and to understand how these microRNA may regulate inflammation, reducing, but also they have other functions. They regulate the proliferation of astrocytes, but also may influence the differentiation of neurostem cells. And this is a microRNA expressed not only in the brain of TSC patients, but also in a large spectrum of pathology. So also in acquired type of epilepsy. And this brings me from the data from the human data to the animal model and established in collaboration with Anna Maria Vezzani. We looked, for example, what is the consequence of the modulation of this specific microRNA 146. So if you increase the level in experimental model, you can interfere with neuroinflammation, hyperexcitability, with a final effect as an example of disease modification. So I hope that I convince you that using human tissue increase the translational value of the experimental models in vivo, in vitro. And clearly, if we want to collect, if we want to use this high quality material, particularly in our case, for my interest, in severe pediatric epilepsy, we cannot do alone in a single center. We need to do an international research network. I think it's really important to consider what I say when I start, the active, early, and sustained involvement of patients and public representatives in biobanking, which is really important. And then the value of the knowledge and neuropathological diagnostic criteria that we have to harmonize, particularly when we are talking about a rare disease. And clearly, we are dealing also with important, complex ethical and legal issues which require refinement. So to conclude, I think the long-term sustainability of tissue collection, and I'm talking particularly in rare disease, is a major challenge. And for these reasons, I think we need clear long-term investment to maintain, to optimize the potential of tissue biobanking. And also colleagues already mentioned, this can be done only within an harmonized network. And I would like to acknowledge colleagues of EpiStop, the collaborators within the neuropathology group in Amsterdam, but also all the collaborators during these 5 years of other consortia that interact with EpiStop, so from EpiTarget, EpiPeer, and Desire, starting from EpiStop data. Thank you.
Thank you very much, Eleonora. So our last speaker for this session is Asla Pitkanen, who will be speaking on behalf of EpiTarget.
Okay, thank you. So we have been hearing about the human databases, so let's now move to the animal databases, because this is clearly something what is new and what is near future. Yeah, sorry if I... yeah, not red one. Oh. Okay. This is the last talk today. Second last. Okay. So when we think about the preclinical studies, so we hear it in many, many places by many, many authorities saying that the preclinical studies are underpowered and not reproducible. And this is a very serious concern that we hear. So what we did in EpiTarget, we tried to address this issue, how to make or do preclinical experiments better. So if we think about currently how the animal records are kept, so it typically looks like this. So we have 1 to 2 year fundings to hire a postdoc or a PhD student or a graduate student, and this is our archive. And then 2, 3 years later, we try to figure out what was done by whom and when. In clinic, of course, it has been much more organized, and what it tends to be in these days is that they are electronic, our patient records are electronic. So to do or to make progress, we were wondering what to do, and we were thinking, how did the clinicians solve the problem? And what we have heard today, so it doesn't seem to be as straightforward on the human side either. But basically, here is the NINDS Common Data Elements webpage. This is a little bit old, it changes, it's very plastic because it renews itself almost monthly. But this is a very interesting and valuable source of information. You can see there that there is epilepsy common data elements. So here is the list of diseases from which there are clinical common data elements. You can actually download the common data elements and case report forms from there, and also find guidelines how to use them. So what we did, so just to summarize, this has come up already a couple of times. What is a case report form? So it's basically built on modules. For example, here is the module on disease or injury-related events. And then here is, for example, classification of seizures. And here you can see the common data element details, kind of the data dictionary. So what we did in EpiTarget, we basically used that as a template to develop the preclinical common data elements and case report forms. We had 8 modules, starting from core animal characteristics all the way to the functional outcome. We had 30 altogether, in the beginning 31 case report forms. We had over 1,000 data points per animal, and then guidelines for each procedure. I can just mention that, for example, in my lab we have been using this now for 3-4 years, and currently we collect about 16,000 data points from each animal. And if you are interested, you can actually go and look at this on our webpage, EpiTarget webpage. So this is an example. This is the core animal characteristics. Basically, this is how we describe our subjects. So we have the animal ID, date of birth, arrival date to the facility, quarantine, identification, etc., etc. So the way how this works, thanks to the help of Roland, who spoke earlier, we actually started to use Redcap. It's in Lund University. Merab is, our coordinator is there. They kindly arranged an international access to the database. So basically what happens, we generate the data, put it on Excel sheets, download to the Redcap, where we have the common data elements and case report forms. It's stored on server, and when we need to analyze, we can pull down the data back to our computers if we wish so. So the computing space is still in each lab. So we kind of prepared the first templates, but this is something what is happening also in other fields in preclinical research. Traumatic brain injury, they have a collection of case report forms and common data elements already. Spinal cord injury is working on those. And then in epilepsy field, so the ILAE, AES, NINDS task 3 has been very active in generating or developing the case report forms and common data elements for different purposes of preclinical epilepsy research. And actually, this was our publication initially, and you can see here all the players who actually contributed to generation of this initial version of common data elements and case report forms. And here is this task 3 publication. There's another one in Epilepsy Open that is in press that also contains the case report forms if you are interested, so you can download them there. So what are the bottlenecks? I think that I'm repeating very much what has been mentioned earlier, what Katja already started, and Roland continued, and others. One of the bottlenecks is that, okay, we are neuroscientists. We are not used to that much with the kind of working with the computers. I mean, what is inside the computers. So we need special kind of IT expertise. This means certainly extra costs. We need to maintain and continue the databases after the project ends. So what is the exit strategy? It's very plastic. We need to do revisions on CDS and CRFs and guidelines. And then, of course, in each case, when one pulls out from the library the case report forms, one has to decide what is relevant. The other thing is, what is a consensus of guidelines? We really need expert panels, so it takes some time. And then one of the biggest bottlenecks is the motivation of the researchers to use these electronic databases. And this is a typical response when I give this talk that not me, I don't want to use this. Why should I use this? So one should be relaxed. This is not for all purposes. This is mostly for example, preclinical treatment trials or preclinical multicenter biomarker studies. And then big issue is data sharing. Do we really want to share? We say that we are sharing, but are we really doing it? And what are the kind of the conditions? So how to motivate? Certainly the funding agencies can influence, the journals can influence us if they require use of standardized procedures in multicenter trials. And then I think Brian Litt suggested that maybe we should have some kind of sharing index so that if the data is good, it's shared, people like to use it. So perhaps this would be one motivator in our CVs. And then really it's very important to keep in mind that this is for well-defined purposes, not for all experiments. So the goal is not to cut creativity, but kind of increase the statistical power of the experiments by harmonization of multicenter trials in preclinical setup. So future directions, I think what I have been hearing here all day, and particularly in this session, and what I think Janet will continue where I stop here. You have this epilepsy mission in your bag, and the way how I see this data bank issue going on is that we really aim to develop the European epilepsy data ecosystem where preclinical data sets will be one component of it, in addition to clinical data sets and probably, hopefully, biobanks, depending on how it will be designed. But this is something I think what seems to be critical to not to waste money, but take full use of the data collected. Thank you very much.
Thank you, Asla. Perhaps I can invite the speakers to come up and then we can have questions. So, any questions? I have a question. You may have feared this would be a dry end to a long day, but in fact I think it shows really the power of biobanking and sharing. And so one question I have is some might argue that we have enough data now, we've reached peak data, all we need to do is share. Do you think that's right, or do you think that we still need to acquire more data? It's just a provocative statement for the end of the day.
That requires a very strong answer. I think we are far away from having enough data. So what I saw here in the genetics discussion always was talking about blood. And as a pathologist, and I really deeply believe that is not equal to what we see in the brain and what we have to treat in the brain, because the brain has probably its own genome. You don't have to forget, it's a post-mitotic organ like the heart. So whatever is acquired during these, let's say, 30 cell cycles that a neuroblast needs to migrate and develop a cortex, can acquire a second hit. So that's all my message to the what is that, AP-stop, TSC group. Don't bother too much about the blood TSC mutation. Go into the tissue and look for the second hit. And that's what we don't have. I mean, we have the tissue, but we need to really look into that more careful.
So Ingmar, you gave the answer I hoped you would.
We already did. I think you can make a comment.
I agree, we should look into the brain more deeply.
For the second hit, he also looked. It's difficult to find, as it's difficult to find also in focal cortical dysplasia. So it's a challenge, but it's a technical challenge. And what I would like to add related to my talk, I think not a lot of people, they understand how difficult it is to collect tissue for rare disease, pediatric rare disease. Also the brain bank, I talk with Netherlands Brain Bank, they say, oh, without extra money, Now, we cannot include in the program the rare disease because for us there is not a big request, and we need at least 20 years to have enough material to say that we have a collection. So there is, I think, we need some support, but a long-term support, and I think this material is of great value because it cannot be resected. Not everything can be resected, and maybe in the future the patients will be treated with good anti-epileptic drugs. They will not undergo surgery. So the only way to get material to understand the link between experimental will be post-mortem material. So I know that not a lot of people they use, but they use always as control. And also there is really important to get a good control, good quality control, age-matched, which is extremely difficult when we are talking about developmental disorder because we are in a really critical age where you have not a lot of material. Okay.
So true. And then even we have to reach the next level. And I think, Eleonora, I thought you would talk about that as well, the single cell level. So we talk about neurons, glial cells, we have different compartments, and they probably contribute differently. And that is what we are not even have a clue how to look at that. But with the innovation in technology over the last 5 years, I think we are going there. We have now single drop sequencing methodology. It's just still very, very expensive. So I can just recall when some consortia was set up and they were funded with 10 million euros, we will have a new disease classification in 5 years. I think that's a reasonable prediction. And we need really to look into that.
And I would just add to that, actually, not just pediatric brain tissue, but adult brain tissue. Because of course, you know, you see what happens in the long term. So there may be pediatric onset. Yeah,
exactly. Pediatric onset, when we are talking, we discuss this problem issue to collect tissue from rare disease. The idea was not to collect pediatric because we hope that the patients with all the treatment, they reach the adult age. The problem that you need really an infrastructure within a brain bank, if you want to do within a brain bank, to follow these patients, to ask for permission, because these are patients with intellectual disability. Then there are ethical issues, there are legal issues. So you have to obtain a donation from from the parents or somebody else who will take care of these patients, which will reach the adult age, but they will die as everyone.
Sorry. Not only the pediatric brain, but exactly also adult brain, the same brain.
Okay, questions from the floor. At the back, please.
So don't you think that this problem of getting post-mortem tissues and getting, more samples of this kind could be overcome involving patients and their representatives in the governance of the biobanks or the registries? I was wondering if you might consider the patient community or their representatives as partners in this kind of infrastructures, also for their long-term development and, maintenance.
I mentioned also in my presentation, active participation of patients and family of patients is the only way if you want to get this type of biobanking. So make really part of the donation.
Yes, with an involvement also in the governance of the infrastructure. Otherwise, it's just donating samples, which is one-way relationship.
As a pathologist like Eleonora and me, we have not direct contact with the patients. So that's why we cannot really interfere here directly. But as an interdisciplinary network, the physician, the neurologist, the treating neurologist should be the person who bridge that gap.
And I can say actually in the UK, the Epilepsy Society Brain and Tissue Bank does have patient representation on the strategic committee. So it does happen. Any other questions from the floor? At the edge there.
So I would actually like to ask Rollin Krauser, because so far we talked mainly, about the tissue side, more or less, of this biobanking. And our experience is that with respect to the tissue samples we have, what's more and more challenging is the IT side, actually to integrate clinical data, which is a real problem to get reconsenting of patients for new studies, things like that. And my experience is, at least what I know from, let's say, German universities, every university is trying, to come up with a individual solution of biobanking and things like that for the same problems everywhere, more or less. So are there comprehensive solutions, or how do you see that?
No, I mean, I see the Informatik Initiative, for instance, which is a German initiative for data sharing between the hospital and the research side, and there are no standard solutions. Everything is Hello Kitty stickers and duct tape. It's
But there are some really innovative initiatives, and also I think they are funded by the European Union. This is, I said it's kind of this MyCloud solution. So the patient has like a device, smartphone or whatever, and he's collecting all the data gathered during the whole epilepsy career, and with this wearable EEG, whatever recordings, they can, feed that. And then if this person is somehow connected to that consortium, you ask, can we retrieve the data? And then they just push the button and say yes, and that's one. I mean, it's far from being developed, but I know at the University of Erlangen we have a group developing the IT solution for that. I think that's very smart, and that's the future.
I would like to add to that there are a few countries in Europe who are taking this GDPR element about patient actually owning their own data very seriously. So there are many places where this kind of citizen folder is about to be implemented. And the whole idea is that there's going to be a kind of a marketplace around that folder structure, so that once a person is in actual real control over their data, they can, for example, receive invitations for clinical trials with all the explanations through their cell phone or something like that. So I would see that patient organizations, stakeholders of that kind would be quite interested in evolving something around that kind of structure. So I think in Europe we are in a quite a good position because of this.
But this is going to be very interesting. I mean, you know the crisis better than I do, the crisis of consent. Do people actually read what they've done? Now, how much are they going to be in control of their complex patient history that they have sitting on their USB key or your smartphone that they might leave in the pub, or, I mean, the CIA director has lost his laptop in the cab, right? This happens. I mean, and that's probably maybe somebody who takes care of this. So I think not all of this is sorted out yet. I mean, there's going to be some transition period where we have to work with solutions that are a bit more mundane. So.
Yeah, but we have to keep the vision.
That's your job, yes.
Follow.
Unless there's a... Sorry. So thank you very much for a great session.